Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullah. Welcome to the seventh lecture of the immunology course, and it's going to be about innate immunity. So the objectives for this lecture, we're going to discuss the concept of innate immunity, features, and important aspects about innate immunity. Also, we're going to explain how the innate immune system recognizes foreign antigens in general. Also, we're going to outline the components of the innate immune system, and we're going to discuss how these components combat various foreign antigens. As an introduction, we'll start with the protection against infections that relies on the mechanism that exists before infection are capable of rapid response to pathogens. So this is the innate immunity. Innate immunity is the first line of defense against infection, and its characteristics are set up at birth. They are non-specific and they are hereditable. There is no immune memory and little individual difference. It's non-specific. Every time the human body encounters the same antigen, it's going to react in exactly the same way. There is no rapid response with second or third exposure because this is non-specific. With the adaptive immune system, there is a memory, so there is faster response upon second, third, or fourth encounter of the same antigen. First line of defense, we have the intact skin, we have the mucous membrane and the secretions, and also we have the normal microbiota. Second line of defense, we have the immune cells such as the natural killer cells and the phagocytic white blood cells. Also, we have inflammation, fever, and antimicrobial substances. And we're going to talk in some detail about these different aspects of the innate immune system in the next slide. So the innate host defense mechanisms, we have the physical factors, biochemical factors, microbiological factors, fever, innate immune cells, cytokines, complement system, inflammation. And we're going to talk briefly about each one of those components of the innate immune system in the coming slides. So for the physical factors, we have the skin. Microbes sloughed off along with the skin cells. Microbes must penetrate several layers. So if there's any breach of the skin barrier, the microbes can get into the body through this breach. So we have stratified and conified epithelium provides a mechanical barrier in the skin. We have indigenous microbial flora competes with pathogens on the surface of the skin. We have microbial flora, and this microbial flora or microbiota plays a role in competing with pathogenic bacteria and preventing it from getting into our bodies. Also, we have acidic pH inhibits the growth of disease-producing bacteria, and we have bactericidal long-chain fatty acids in sebaceous gland secretions, also which plays a role in protecting our body from the invading pathogens. Mucous membrane also produces mucus to trap microbes, and it is mostly lined with cilia. So presence of mucus and the movement of the ciliated epithelium works in favor of getting out any foreign pathogens. Biochemical factors, we have the low pH in the vagina, urinary tract, and the stomach, and this low pH plays a role in protecting these sites within the body or organs within the body. Also, we have defensins, which are short antimicrobial peptides insert into bacterial membrane and form pores. We have defensins, short antimicrobial peptides. and these play a role in pore formation within the bacterial membranes if they invade the human body. Also, we have the lysozyme, which degrades peptidoglycan. Tears contain high concentration of lysozyme, and it is effective against gram-positive microorganism. Also, we have interferon, which is a cytokine that triggers macrophage activation and produce a substance to interfere with RNA viral reproduction. رح نحكي في السلايدات القادمة عن the role of the cytokines and the role of interferon as part of the cytokines, and we have already covered the role of interferon previously when we talked about the innate immunity, when we talked about the immune system in the virology course. So the antimicrobial peptides, or the defensins. Defensins originally isolated from frog skin based on their ability to kill bacteria. They first were identified in the frog skin, and they were found to kill bacteria. 400 peptides are described up to date. Defensins are grouped into 4 families in the eukaryotes. We have the alpha and beta defensins, which are present in the humans, and we have the insect defensin and the plant defensins. In the humans, the alpha defensins are mostly within the neutrophils and the intestinal Paneth cells, and the beta defensins are within the epithelial cells. The defensins appear to act by binding to outer membrane of bacteria, resulting in increased membrane permeability. So once they bind to the outer membrane of the bacteria, they increase the membrane permeability of the bacteria and leads to the influx of fluids inside and leads to death and lysis of the bacterial cell. Classified based on their secondary structural features. So we have those which are composed of alpha helical peptides, and we call those cathelicidins, and we have the beta strand peptides, which we call them defensins. The human cathelicidins, we have the LL37, is highly expressed by polymorphonuclear cells and numerous mucosal and epithelial cell types. The beta strands of the defensins are connected by disulfide bonds, and the defensins mostly are short peptides, less than 100 amino acid, and they carry a positive charge. They interact with microbial cell membrane component to increase cellular permeability, resulting in cell death. They also play a role in modulating the inflammatory process and play a role in the wound healing and repair. Third innate defense mechanism is the microbiological barrier. So we have the normal flora or the normal microbiota. They are not part of the immune system, but they play a role in the defense against pathogenic bacteria. And protection they provide is considerable. They have 3 mechanisms of action. So it is competitive exclusion of invading microbes. They compete with the invading microbes for resources. They produce compounds that are toxic to other bacteria. So their presence in our bodies as a defense mechanism in the environment that they're living with, they produce toxic materials. If any pathogenic bacteria comes, then these pathogenic bacteria would face these toxic materials and would not survive within this environment. Also, they stimulate immune system. So their presence within the human body is going to stimulate the immune system. So it's going to be presented by antigen presenting cells, or the immune system is going to be triggered by the antigens of these microbiota. But as we said previously, for microbiota, the immune system has the ability to recognize them as self or as non-harmful, and thus it's not going to produce any immune response against it. So من هذا المنطلق بنحكي هو بيعمل just stimulation، تحفيز، تدريب لجهاز المناعة، but it's not going to activate specific immunity against these microbiota. The fourth mechanism is fever. Fever results from the release of pyrogens such as interleukin-1 and interferons, and also from toxins from the infectious agents, from drug reactions, from brain tumors. And the pyrogens which are released and circulate through the body, they are going to target the hypothalamus and cause release of prostaglandin E2. The release of prostaglandin E2 is going to raise the temperature set point of the hypothalamus. وبالتالي عندنا الهايبوثالاموس فيه زي thermometer، زي sensor set to normal temperature. Once there is presence of infectious agents, drug reaction toxins, brain tumors، بصير عندنا production of prostaglandin E2. The prostaglandin E2 is going to lead to setting the hypothalamus at a higher temperature. What are the benefits of fever? Inhibits reproduction of bacteria and virus. So at higher temperature than the normal body temperature, this is going to inhibit the reproduction of bacteria and viruses. It's going to promote interferon activity, which plays an important role against the reproduction or the replication, especially of viruses. Increase the activity of the adaptive immunity. As we always say, the innate immune system and the adaptive immune system work together and in collaboration. So one of the triggers for the adaptive immunity to become activated is via fever, accelerate tissue repair, and increase the CAMs on the endothelium of capillaries and lymph nodes, leads to additional immune cell migrating out of the cell. زي ما حكينا عن الICAM، حكينا التريجرز اللي هو the production of interleukin and interferon by macrophages في ال site of inflammation. الجسم بيشتغل كله مع بعض. جزء من اللي راح يصير انه راح يصير في عندنا release of prostaglandin, increase in the temperature، وبالتالي شو بده يصير في عندنا increase of the production of the ICAM على ال inside of the endothelium، وبالتالي this is going to lead to white cell extravasation and migrating out of the blood into the affected tissue. Recommended to leave a low fever untreated. But if we have high grade fever, it needs to be treated because it might cause harm more than benefit. Risk of high fever, significant above 100 degrees Fahrenheit. High fever potentially dangerous above 103 degrees Fahrenheit, especially in children, even in adults. A change in metabolic pathway and denaturation of protein at these high temperatures, and it might lead to seizure, irreversible brain damage, and maybe death if the fever is too high. Innate immune cells that play a role, we have the monocyte macrophages, and their principal function is phagocytosis, inflammation, T-cell activation, and tissue repair. So for monocytes and macrophages, حكينا monocytes, they circulate in the blood for up to 8 hours. After that, they bind to ICAMs، حكينا الICAM2 expressed below levels على الاندوثيليوم of the vessels، وبالتالي بعد جولتها 8 ساعات they can go to the tissues to become macrophages. So they play a role in phagocytosis, inflammation, T-cell activation, and tissue repair. We have the neutrophils, which has a role in phagocytosis and inflammation. We have the natural killer cells, which from its name, the function is killing of infected cells, virally infected or intracellular bacteria infected cells, and tumor cells. We have dendritic cells, which play a role in phagocytosis and activation of the naive T cell. We can see that the dendritic cells, the macrophages, they are professional antigen presenting cells, so they play a role in activation of the naive T cell. And also we have the mast cell that produce histamine, and it mediates inflammation. Also, we have eosinophils, which has the ability to defend the human body against parasites. Phagocytes performed by neutrophils and macrophages. Phagocytosis is the capture and digestion of foreign particles. So if we have foreign pathogen, it's going to be engulfed and captured, and then these phagocytic cells are going to start digesting that foreign antigens. Chemokines are cytokines that attract macrophages and neutrophils to infected tissue. Also, they are going to produce interleukins and tumor necrosis factors that are going to serve as chemokines or chemoattractants for recruitment of other macrophages and neutrophils to come to the site of infection. Also, it is going to work with opsonins. Opsonins attached to microbes to increase the ability of the phagocytes to adhere. So opsonization make it easier for phagocytic cells to target these cells. Opsonization حكينا عنه أكثر من مرة، ولازم تكونوا عارفينه، اللي مش عارفه يروح يراجع شو يعني نقصد ب opsonization. So steps of phagocytosis: the pathogenic or the foreign antigen is recognized first, then it is ingested via the pseudopods, which engulf the microbe through endocytosis. Then a vacuole is going to form, and the vacuole is going to have or contain the microbe or the pathogen. Then it is going to be digested through the vacuole is going to merge with enzymes to destroy the microbe, and then the microbial debris is released and exocytosed. So these are the steps that we talked about. Formation of pseudopods. This is the microbe or the pathogen or the foreign antigen, and this is the phagocytic cell. It's going to form the pseudopods اللي هي الذراعات هاي, and then it's going to engulf the foreign antigen and ingest it into the phagocytic vesicle. Then the phagocytic vesicle is going to, or the lysosome is going to fuse with the phagocytic vesicle, forming what is called phagolysosome. And the phagolysosome or these digestive enzymes are going to destruct and destroy and kill the invading pathogen. And then after that, the formation of residual body containing indigestible material is going to occur, and this next step is discharge of the waste material outside of the phagocytic vesicle. So innate immune recognition, how does the innate immunity or the innate immune cells recognize that this invading pathogen is non-self? All multicellular organisms are able to recognize and eliminate pathogens. Despite their extreme heterogeneity, pathogens share highly conserved molecule called pathogen-associated molecular patterns. وبالتالي الفيروسز، البكتيريا، الباراسايت، whatever، في عندنا heterogeneity ويعني اختلافات عظيمة في السيكونسز وفي الستراكشر. Despite that, all these foreign antigens, they have what is called pathogen-associated molecular patterns. The pathogen-associated molecular pattern is the part of these foreign antigens or pathogens that is being recognized by the body. ليش؟ لأنه الPAMPs اللي بنسميهم pathogen-associated molecular pattern or PAMPs, they are unique for bacteria, viruses, or whatever, and they are not present within our bodies. وبالتالي مجرد ما تدخل, it is going to be recognized as foreign. Host cells do not share PAMPs with pathogens. هاي السيكونسز هي خاصة بالبكتيريا أو الفيروسز أو الفورين أنتيجنز. هاي السيكونسز مش موجودة بأجسامنا، وبالتالي جهاز المناعة مجرد والخلايا مجرد ما تعرف عليها, it's going to recognize that this is foreign. PAMPs are recognized by innate immune recognition receptors called pattern recognition molecules or receptors. كيف راح يتعرف عليها؟ إذا في عنا pathogen associated molecular patterns PAMPs، أو في عنا DAMPs كمان, damage associated molecular patterns, which results from tissue damage or injury to the tissues. These are going to bind to pattern recognition molecule on the surface of the innate immune cells. What are the typical PAMPs? What are they made of? Lipopolysaccharide, peptidoglycan, a nucleotide sequence unique to bacteria, and some other bacterial components. زي ما حكينا, these sequences are unique for these pathogens. They do not exist in our bodies, so they are going to be recognized. So PAMPs or DAMPs. So this is the non-specific immune cell. Once they are recognized, they are going to bind to, as we said, the pathogen recognition receptor. Binding of innate immune receptors and PAMPs are going to mediate inflammatory cytokines, or they are going to lead to antigen presenting cell recognizing PAMPs. This figure was taken from as a screenshot from one of the YouTube videos talking about the innate immune system and the PAMPs and the pathogen recognition receptors. So what is the fate of the pathogen once recognized by the pathogen recognition receptor? We have three types of pathogen recognition receptor. We have extracellular, we have intracellular, and we have secreted or free pathogen recognition receptor. The intracellular lies within the membrane of the macrophage. This is a macrophage. So once the pathogen, the PAMP of the pathogen is recognized, it's going to bind to the pathogen recognition receptor, and it's going to internalize or engulf this foreign pathogen, and it's going to lead to either engulfment and killing of the pathogen within the phagosome lysosome, and the pathogen recognition receptor in this case is the mannose receptor or the scavenger receptor, or it is going to lead to the binding of the PAMP with the pathogen recognition receptor, or the PRR, is going to lead to sending a signal to activate genes within the cell, and these are going to lead to release of cytokines such as interferon. And this PRR receptor is, in this case, it is the Toll-like receptor or the signaling receptor. Also, we have the case of intracellular inside the cell. If the pathogen recognition receptor is inside the cell and the pathogen is a virus or an intracellular bacteria, it is either going to lead to release of cytokine, and in this case, when the pathogen is intracellular, the receptor is NOD-like receptor. ما راح نحكي بالتفاصيل عن انواع الريسبتورز، المانوز ريسبتور، سكافنجر، التول لايك، النود لايك ريسبتور، في تفاصيل كثيره اذا بدنا نيجي نبحث في الشغلات هاي. خلينا بس على التفاصيل اللي عم بعطيكم اياها. او في حاله انه the cell, the macrophage here we're talking, if it became infected it might lead to death. So if the pathogen binds to the PRR, then this is going to lead to activation of the signaling cascade, which is going to lead to release of cytokine, and in this case the PRR is NOD-like receptor. In the some immune cells also secrete these PRR receptors, which are complementary receptors, and these receptors are going to, in this case, are going to help the complement system or the complement cascade in its activation process. So we talked about the TLRs. TLRs, we have multiple types of TLRs. TLRs are a type of pattern recognition receptor. So we have the TLR4 here, for example. It binds to gram-negative bacteria. TLR5, for example, recognizes bacterial flagellin. TLR3, 4, 7, 8, all they recognize the viral, either DNA in the case of 3, or 7 and 8 single-stranded RNA, or the TLR4 viral proteins. And those are extracellular, those are intracellular type of pathogen recognition receptors. The sixth component of the innate immunity is the cytokines. Cytokines are a broad and loose category of small proteins that are important in cell signaling. They play an important role in cell signaling, and their release has an effect on the behavior of cells around them. In response to microbes, macrophages and other cells secrete proteins called cytokines that mediate many cellular reactions in innate immunity. So, as a result of presence of microbe, the innate immune cells are going to produce cytokines. These cytokines are released from either this cell to affect and protect the same cell, or to protect nearby cells, or even sometimes to protect distant cells. Cytokines are produced by a broad range of cells, including immune cells like macrophages, B lymphocyte, T lymphocyte, and mast cell, as well as endothelial cells, fibroblast, and stromal cells. Cytokines play a role in being an inflammatory mediators of inflammation, and they play a role in communication between leukocytes and other cells. And as we're going to see later, cytokines play an important role in activation of the specific immunity. So the specific immune components, they are going to be activated and triggered by cytokines. راح نشوف بالرسمة اللي في آخر المحاضرة. We have 4 types of cytokines. We have chemokines, and chemokines, they are important in chemotaxis of immune cells. So there is a site of injury, there is a site of inflammation, there is a site of infection, and we need to recruit more cells to target that location. The chemokines of the cytokines play a role in recruiting and guiding these cells to the site of infection. We have interferons, which are glycoproteins important in the control of viral infection, also help regulate cell involved in the immune response. So we have chemokines, interferons, interleukins, and tumor necrosis factors, the cytokines. Interleukins important in innate immunity, inflammation, adaptive immunity, and the vast majority of these are produced by the helper T cell, as we're going to see in the last slide of this lecture. Tumor necrosis factor help kill tumor cells, initiate programmed cell death, apoptosis in infected and cancerous cells. So functional features of the cytokines, they are potent. Their effect is local. So we have the local is divided into autocrine, paracrine, and endocrine. Autocrine is that the cytokine is released by the same cell to protect and help the same cell that that has released the cytokine. Paracrine is cytokines produced by this cell to warn and protect the neighboring cells. And sometimes if these cytokines were produced by a cell and released into the circulation to go to distant cells, it is called endocrine effect. It's highly interactive, pleiotropic, redundant, synergistic, and antagonistic, and we're going to talk about these interactions in the next slide. Functional features, potent, so the inducing stimulus is going to lead to activation of the cytokine gene to produce the cytokines from this cell, and it is the cytokine going to bind to the receptor on the nearby cells or the same cell that has released the cytokine. It's going to lead to gene activation of the target cell, and it's going to lead to biological effects, which the end result of it is protection against the invading pathogen. So the local effects, autocrine released by this cell, and then it comes and binds to receptor of the same cell to protect this same cell and activate genes within this cell to protect the cell from invading pathogens. Paracrine, as we said, produced by this cell, and it is recognized by receptor on the nearby cells. And the endocrine, it is released by this cell, go through the circulation to distant cells. So this is the endocrine. Highly interactive, we said pleiotropic. So here we can see this is an activated T helper cell, and the cytokine interleukin 4 act upon this activated T cell, and it might lead to the interleukin 4. So one cytokine might lead to activation of multiple things as a result. So here we see activation, proliferation, differentiation of B cell, and proliferation of thymocyte, and also proliferation of mast cell. This effect is called pleiotropic. cytokine واحد ممكن يعمل more than one function. Here we have redundancy. Redundancy means that interleukin 2, interleukin 4, interleukin 5, all these interleukins might have the same role and does the same thing. So in this case, interleukin 2, 4, or 5 leads to B cell proliferation. طب كيف بتصير هاي؟ كيف الانترلeukin 2 و4 و5 have the same effect? Because interleukins they can share common surface receptors and can activate common cell signaling pathways within these cells. So in this case, we have the activated T helper cell can be activated by interleukin 2 or 4 or 5 to lead to B cell proliferation. أكثر من type of interleukin عم بيعمل نفس الوظيفة هاي بنسميها redundancy. Synergistic effect. Synergistic effect. We have activated T helper cell. It is activated by interleukin 4 and interleukin 5. So this is going to have synergistic effect. In this case, it's going to lead to induces a class switch to IgG. واحنا بنعرف يعني synergistic effect. يعني synergistic effect انه وجود التو interleukins راح يعطونا قوة مضاعفة او حتى مش مضاعفة، يعني هي مش مجموعهم مع بعض، لا هو اقوى من مجموعهم، وبالتالي في هاي الحالة it led to class switch to IgE. We have antagonistic effect انه واحد بيشتغل عكس الثاني. So the interleukin 4 هون حكينا it induces class switch to IgE، بينما الانترفيرون gamma antagonizes this effect by blocking the class switch to IgG induced by interleukin 4. synergistic اللي هو بعطينا قوى مضاعفة، antagonistic عكسي، redundancy إنه more than one cytokine can do the same action on the cell, and pleiotropic إنه one cytokine can do multiple actions. Cytokine families, we have the hematopoietic family, interferon family, tumor necrosis factor family, and the chemokine family. As we said previously, 1, 2, and 3 elicit physiological response, whereas 4 serve as chemoattractant and chemo has chemotactic effects. So the hematopoietic cytokines, these are examples of the hematopoietic cytokines. مش مطلوب منكم تحفظوهم، بس على الأقل اطلعوا على السلايد تمعنوا، حاولوا تشوفوا differences between the different families. So this is the interferon class 2 cytokine receptor, interferon alpha, beta, gamma, and interleukin 10. Here we have the tumor necrosis factor receptors, tumor necrosis factor alpha, beta, CD40, nerve growth factor, and the FAS. And we have the chemokine receptor, interleukin 8, RANTES, MEP1, PF4, MCAF, and the NAP2. So what does interferon do, especially in viral infected cells? So this is an infected cell. It leads to the production of the cytokine, which is the interferon, where it is going to be picked by receptors on a normal cell. It is going to lead to synthesis of enzymes that interfere with the viral replication. Also, it's going to trigger the natural killer cells to target virally infected cell, produce perforins, granzymes, and leading to death and apoptosis of the virally infected cell. And also, it is going to trigger and activate macrophages to phagocytose and engulf the virally infected cell into an endocytic vesicle, and then via phagosome, lysosome is going to lead to destruction of the invading pathogen and the infected cell. So it binds receptor of neighboring cell, promote macrophage function, apoptosis of infected cell, trigger synthesis of enzymes destroying viral RNA or DNA, and trigger synthesis of enzyme that inhibit synthesis of viral protein as well. So in this figure, if we look at the this figure, we can see that this is the fibroblast, endothelial cells, and this is the macrophage. So once the macrophage senses the presence of foreign pathogen, we said it's going to produce interleukin-1, tumor necrosis factor. When we talked about the endothelium and the white blood cell extravasation, also it's going in the T helper cell. This is the naive or the resting T helper cell. Once it is activated, we can look at the amount of the interleukin or the cytokine mediators that activate other cells, such as the natural killer cells, the LAC cell, the T cytotoxic cell, the activation of the B cell through the helper T cell, also activation of the B cell activation of the plasma cell to produce antibodies. So cytokines play an important role in the innate and the adaptive immune response. The sixth component is the complement system. The complement system is a collection of circulating and membrane-associated proteins that are important in defense against microbes. The complement system proteins are all synthesized in the liver, and they are released into the circulation. Many complement proteins are photolytic enzymes, and complement activation involves the sequential activation of these enzymes called enzymatic cascade. بنحكي complement system أو complement cascade. لأنه لما نحكي عن complement system أو complement cascade في عندك انزيم راح يتم تصنيعه، وبالتالي راح يرتبط مع الانزيم أو البروتين اللي قبله، بيعمل له activation، وبعدين مجموعهم مع بعض is going to activate the next step. The next step is going to depend on activation of this new enzyme, and as we said, so it is a cascade. لازم تعمل activation لواحد حتى يشتغل على اللي بعده، تعمل اللي بعده بنعمل له activation حتى يشتغل على اللي بعده، وبالتالي هم مترابطات بحاجة إلى activation الخطوة السابقة حتى الخطوة اللاحقة تمشي. مشان هيك بنسميه complement cascade. We have three pathways to activate the complement system. We have the classical, activated by antibody binding to microbes or antigen. We have the alternative, directly activated by microbes. إذا الكلاسيكال activated by antigen antibody reaction. لما نحكي عن classical complement system، وحكيناها قبل هالمرة لما حكينا عن الكومبلمنت activation. بالكلاسيكال it's triggered by antigen antibody complex. بالتالي في عندنا antigen antibody complex. This is going to trigger the activation of the classical pathway. The alternative pathway is directly activated by the microbes. وفي عندنا lectin pathway binds to mannose containing carbohydrates. في عندنا بعض أنواع البكتيريا زي النيسيريا، السالمونيلا، الفنجاي زي الكانديدا، في عندنا فيهم مانوز، وبالتالي these bacteria trigger the lectin pathway and it does not require presence of antibodies. Host cells have complement regulatory proteins on their surface that protect them from spontaneous activation of C3 molecule, while microbes can activate the complement pathway, but it have no regulatory proteins. هلا رح نعرف ونحكي إنه C3 هو جزء مهم من الكومبلمنت سيستم، وبالتالي الهوست سيل في عندها regulatory proteins تمنع الactivation of the C3، بس إذا في عندنا foreign antigens they are going to be targeted by the proteins of the complement system. When pathogen activates the complement system, this initiates innate immune response by three main mechanisms. The final outcome of the complement activation is trigger of inflammatory response, lysis of the invading cell or pathogen, and opsonization by the components of the complement system in order to be targeted by the phagocytic cells. So here we have the classical pathway. The classical pathway, as we said, it is triggered by antigen-antibody reaction. Antigen-antibody reaction is going to activate the C1 component of the classical pathway. The C1 is going to activate the C4 protein of the classical pathway. This is going to lead to C4 giving two products, C4A and C4B. Usually لما نحكي A و B, A is the small fragment, B is the large fragment. Exception is C2. C2, when we say A is the large fragment, B is the small fragment. So C1 activates C4 to give C4A and C4B. Then C1 binds with C4B. هاي اللي سميناها الكاسكيد. 1 activated 4. بعدين 1 ارتبط مع 4B. وال C1-4B activated C2 وأعطانا ال C2 C2A and C2B. Then C2A was bound with C1-4B to give what is called C3 convertase. اللي هي C3 convertase بنسميها C1-4B 2A. هاي هي C3 convertase. وبالتالي كل الخطوات هاي صارت في cascade. This activated this gave two products. Then this, the C1 ارتبط مع C4B، عملنا activation لل C2، أعطانا C2A C2B. C2A ارتبط مع C1-4B 2A اللي هو صار اسمه C3 convertase. ال C3 convertase ارتبط مع مين؟ C3 convertase أثر على مين أو activated مين؟ C3. أعطانا C3A و C3B. C3A حكينا اللي هي small fragment of the C3 and the C3B is the large fragment of the. هلا C3B ارتبط مع C1-4B 2A 3B. أعطانا this product gave us the C5 convertase. ال C5 convertase على مين راح يأثر؟ راح يأثر على C5. شو راح يعطينا؟ راح يعطينا C5A و C5B. ال C5A C3A C4A هذولا ما إلهم دور في الكلاسيكال باثوي. راح نحكي عن الدور تبعهم هون. هون بيجي الدور لمين؟ لل C5B. C5B ارتبط مع مين؟ مع C6 والC7 والC8 والC9. عمل لنا شيء اسمه membrane attack complex اللي هو C5B 6 7 8 9. الممبرين attack complex اللي هو بيعمل لنا زي channel within the invading pathogen. السيل wall of the invading pathogen بيعمل فيه زي channel. وبالتالي لما يعمل channel ايش؟ عمله exposed to the outer environment. بصير في عنا influx from the inside to the outside and influx of fluids or water or whatever into the inside, and this is going to lead to death and lysis of the target cell. إذن هاي هي خطوات الكلاسيكال باثوي. نرجع نعيدها على السريع. C1 activated C4 أعطانا C4A و C4B. ال C4B ارتبط مع ال C1 أعطانا C1-4B. ال C1-4B activated ال C2. شو أعطانا؟ ال C2A وال C2B. هلا ال C2A ارتبط مع ال C1-4B 2A كان بولنا اللي هو C3 convertase. C3 convertase أثر على C3 أعطانا C3A و C3B. C3B ارتبط مع ال C3 convertase عملنا C5 convertase. C5 convertase أثر على C5 أعطانا C5A و C5B اللي ارتبطوا مع C6 C7 C8 C9 وكونوا الممبرين أتاك كومبلكس اللي هو زي ما حكينا بالنهاية بده يعمل لنا زي channel within the membrane of the pathogenic bacteria. طيب شو دور ال C3A وال C5A ما دام ما لهم دور كبير في عملية الactivation of the classical pathway cascade؟ ال C3A وال C5A play a role in the chemotaxis of the immune cells to the site of infection or injury. The C3A, C4A, C5A also they induce acute inflammation by activating mast cells and neutrophils. So this is the classical pathway. We have the alternative pathway. حكينا شو الستيمولس في الالترناتيف pathway. الستيمولس اللي presence of الباثوجين. Presence of pathogen. وبالتالي in the presence of pathogen شو بصير في عنا؟ spontaneous hydrolysis of the C3. بصير في عنا hydrolysis of the C3 بعطينا ايش؟ C3A و C3B. C3B بيرتبط مع في عنا في الالترناتيف باثوي في عنا فاكتور B وفاكتور D. بالاضافه للسي 1 للسي 9. هون بالكلاسيكال باثوي في عندنا from C1 to C9. في الالترناتيف باثوي في عندنا بالاضافه لل C's we have the factor B and the factor D. So بصير في عندنا spontaneous hydrolysis، وزي ما حكينا قبل هيك بالهوست سيلز في عندنا شيء to prevent the activation of the C3 وانه to be targeted للself cells to be targeted by the C3. بال pathogenic bacteria or foreign antigens spontaneous hydrolysis of the C3 بعطينا C3A و C3B. في هاي الحالة C3B راح يرتبط مع الفاكتور B. لا C3B صار له hydrolysis. هلا في عندنا الفاكتور B راح يأثر عليه الفاكتور D، راح يعطينا BA و BB. هلا البي C3B راح يرتبط مع الBB، وبالتالي شو راح يعطينا C3BBB اللي هو C3 convertase، زي C3 convertase اللي هون. بس هون نتج من ايش؟ من في عندنا C3 صار له spontaneous hydrolysis اعطانا C3A و C3B. C3A ما لهوش دور في الالترناتيف باثوي زي ما لهوش دور في الكلاسيكال باثوي في الاكتيفيشن للكومبوننتس اوف ذا باثوي. في عندنا الفاكتور B. الفاكتور B is going to be activated by ايش؟ by factor D. بعطينا BA و BB. هلا C3B راح يرتبط مع الBB، شو راح يعطينا؟ C3BBB اللي هو ايش؟ اللي هو C3 convertase. هلا C3 convertase راح يعمل لنا بعد هيك activation للC5 convertase، وبعدين الC5 راح يعطينا C5A و C5B. الC5B راح يعطينا راح يرتبط مع C6 C7 C8 C9 يعمل الممبرين اتاك كومبلكس. وبالتالي الخطوات البدائيه في الالترناتيف باثوي بتختلف عن الكلاسيكال باثوي، بس بعد C3 convertase راح تكمل نفس الخطوات اللي هو activation C5 convertase، وبعدين انه يعطينا membrane attack complex اللي راح يعمل لنا channel within the pathogenic bacteria let's say and it's going to lead ultimately to lysis and death. The third pathway that we need to talk about is the lectin-mannose pathway. We have the mannose binding lectin أو the MBL. في عنا بعض البكتيريا زي النايسيريا، الكانديدا، الفنجل اللي هو الكانديدا، السالمونيلا البكتيريا، في عندهم mannose وجلوكوز within their structure. So mannose binding lectin is going to bind to mannose in these bacteria, and it's going to recognize the mannose in these bacteria, and it's going to activate the mannose binding lectin associated serine protease أو اللي اسمه MASP اللي هو mannose binding lectin associated serine protease. This MASP is going to bind to the MBL, and it's going to activate ويأثر على ايش؟ على الC4 وعلى الC2. الC4 شو راح يعطينا؟ راح يعطينا C4A و C4B. الC2 راح يعطينا C2A و C2B. هلا راح يرتبط الC4B مع الC2A ويعمل لنا اللي هو الC3 convertase. بعديها الخطوات راح تكمل C5 convertase وبعدين membrane attack complex. وبالتالي كل الخطوات تبدأ في أسلوب مختلف، ولكن بعد الC3 convertase بصير في عندنا activation للC5 convertase، وبعدين الخطوات اللي تعديها حتى يعمل لنا membrane attack complex. وبالمحصلة كلياتها it's going to form a channel within the invading pathogen, and it's going to lead to lysis of the death and lysis of the invading pathogen. C3B بيعملنا opsonization of the invading pathogen، وبالتالي it has a role in opsonization زي ما حكينا. هاي إذا شايفينها هون this is the membrane attack complex اللي هو composed of C5B, C6, C7, C8 and C9. الجدار تبع الشانل اللي هو السي 9. هذا زي ما انتم شايفينه في عندنا تقريبا 18 يونت اللي بتلعب دور انها تكون لنا الجدار تبع الشانل. هذول مكونين من السي 9 والاذر components C5B 6 7 and 8 بتكون within the channel, and the end result is the cytolysis اللي هو lysis of the invading bacteria or whatever. C5A زي ما انتم شايفين هون حكينا it plays a role in inflammation, it affects the mast cell والهستامين. So الستبس من هون كمان مرة ولو انه حكيناها احنا بالتفصيل هناك. step 1 activate C3 splits into C3A and C3B. C3B binds to microbe resulting in opsonization. C3B splits into C5 into C5A and C5B, and C5B bind to C6 through 9 to form membrane attack complex. حتى يمكن احنا حكيناها قبل بشوية تفصيل أكثر من اللي موجود هون في عنا alternative pathway which is triggered حكينا ب presence of the bacteria وبصير في عنا spontaneous hydrolysis of the C3. الclassical pathway antigen antibody complex, وفي lectin mannose pathway في عنا MBL recognizes mannose in certain bacteria and fungi يعمل conversion of the C3 into C3A and C3B. C3B يكون one of the components that leads to the formation of the C5 convertase, وبالتالي بيحول لنا C5 ل C5B اللي بيرتبط مع ال 6 7 8 9 وبعدين بيعمل لنا channel اللي حكينا جداره هون مكون من C9 and the end result is lysis of microbe. C5A inflammation, C3A inflammation, C3B opsonization and phagocytosis. So this is for the components of the innate immunity. In this last slide, we're going to talk about the role of innate immunity in stimulation of adaptive immune response. So adaptive immune system activation of T or B cell need two signals for activation. First signal, antigen recognition. Second signal, derived by innate immunity. زي إيش؟ زي هون عم نحكي عن dendritic cell. Dendritic cell it's an antigen presenting cell. حكينا professional antigen presenting cell. شو رح تعمل؟ once إنها ارتبطت مع البكتيريا وهون رح ترتبط مع مثلا TLR4 or TLR5 اللي حكينا عنهم سابقا. راح يدخل it's going to engulf the invading pathogen وبعدين راح تعملها presentation على MHC وبعدين على MHC it's going to be displayed on the surface of the antigen presenting cell. It's going to be recognized على MHC2 وبالتالي it's going to be recognized by CD4 وبالتالي هاي هي bridging, the role of the dendritic cell in bridging innate immunity to adaptive immunity. هلا تفاصيل adaptive immunity راح نحكي عنها بعدين بس هاي لتوضيح انه innate immunity وخاصة dendritic cell it's going to bridge وتهيئ adaptive immunity من خلالها. يعني الانتيجين اللي موجود سواء exogenous زي البكتيريا اللي موجودة هون أو الفيروس اللي موجود هون و in both cases لأنه this is an antigen presenting cell, in both cases it is being an exogenous the source of the antigen is exogenous سواء بكتيريا ولا فيروس لأنه حكينا dendritic cells it's a professional antigen presenting cell هي part of the innate immunity وبالتالي it's going to engulf any invading pathogen بتدخلوا جواتها تعملوا presentation على MHC class 2 it's going to be recognized by the CD4 والCD4 زي ما حكينا is going to lead to activation of the humoral and the cellular response وبالتالي بداية تحفيز جهاز المناعة المتخصص أو الادابتيف immunity راح يكون عن طريق إيش؟ عن طريق الدندريتك cells أو الإنيت immunity وبالتالي صناعة antibodies متخصصة against this specific antigen أو تصنيع الهيلبر and cytotoxic T cells against this specific antigen مع الميموري cells البي والتي memory cells اللي هي activation of the adaptive immune system اللي راح نحكي بالتفصيل عنه بالمحاضرة القادمة. What's the thing that bridges the gap وبتوصل الانتيجنز ما بين ال innate و specific immunity هي ال professional antigen presenting cells especially the dendritic cells. بهيك بنكون أنهينا المحاضرة اللي بتحكي عن ال innate immunity. So do not hesitate to ask any questions. That's it for today's lecture. وكل عام وأنتم بخير.